third speaker is Dr. Fidel Alfaro. And Dr. Alfaro has acquired his PhD in Oxford in 2020 in clinical neuroscience. And currently his work focuses on developing fully automated analysis tools for the UK Biobank project, which includes 6 different brain imaging modalities acquired from 100,000 subjects. So let's welcome Dr. Alfaro.
Thank you. Well, this talk is going to be a call for ideas for the new version of the UK Biobank pipeline. Okay, version 1 of the UK Biobank pipeline was developed, was released around 8 years ago, and we have already processed more than 72,000 datasets with that pipeline. And more than 500 papers have been published using the data that is generated by the pipeline. It's a set of Bash, Python, and Matlab scripts. The building blocks are mostly FSL and FreeSurfer. And with the image processing and the quality control, we get a set of IDPs and process nifties that are fed back into UK Biobank database. And one of our main goals was to keep backwards compatibility, meaning that we did not change the main functionality of the pipeline to allow for data consistency over time. But we added new features like ASL processing or QSM processing, but the main part of the pipeline is frozen from the beginning. And that's why we are working on BIP, which is, it stands for Brain Imaging Pipeline, which is a new pipeline fully in Python. We want to include the state-of-the-art tools and methods, and we would like it to be usable for projects that are similar to UK Biobank. And that's where we need ideas from the audience. I'm going to describe some of the features that will go into this new version of the pipeline. I will start with the framework, which has 3 basic elements. The first one is FSL Pi, developed by Paul McCarthy, which is a lightweight Python interface to FSL functions. And then there are 2 tools developed by Michiel Kothar, FileTree and FSL Pipe. The first one, FileTree, is a Python library to abstract out file paths and directory structures. And the second one is FSL Pipe, a declarative pipeline framework built on top of FileTree, which allows developers to design new pipelines by specifying the different steps as Python functions, and by specifying the input and output of each function, FSL Pipe automatically links them, and then it creates the pipeline for you. It supports parallel execution and is very efficient that's a very efficient job management. If you want to speak about these tools with Michiel, he will be around poster 2266 from tomorrow. At the heart of the pipeline, there's a Morph, which is FSL's multimodal registration framework, developed by Rick Lantz, which is a new registration tool that allows for simultaneously aligning multiple scalar and tensor images. It has achieved state-of-the-art registration accuracy with biological plausible deformations. Also, the pipeline will now have one warp for all modalities, which is different from what we had before in the pipeline, and it will enable multivariate voxel-wise analysis. This tool is already available in FSL, and yeah, it has allowed us to do this next step, which is we want to move to a different template, the Oxford multimodal template, OMM1, which was developed by Christopher Hofer and Rick Lantz. This template was generated from 240 UKB individuals. The age range was 50 to 55-year-old. Half of them were females. The modalities that were used were T1 weighted, T2 FLAIR, and DTI. And we would like it to be a new standard for multimodal imaging analysis because it will allow for volumetric analysis space across modalities in UK Biobank. And as I said, this is also included in FSL right now. An idea that we are playing with right now is to use age-dependent templates. These have been also generated by Christoph and Rick Lantz, and they are using the same philosophy as the OMM1, using also MMorph as the registration tool. And because as you can see in this video, the ventricles are very different at the beginning of the age range in the UK Biobank and at the end. And these templates have been trained from more than 37,000 UK Biobank individuals. They cover the whole age range from UK Biobank, and they model trajectories of a healthy aging and also remove template age bias. Another tool that we are planning to include in the pipeline is FREDDI, which is EDI for fMRI. Everybody knows EDI. It's a tool for distortion and motion correction of diffusion data, developed by Jesper Andersson. FREDDI performs slice-to-volume motion correction and susceptibility by movement interaction correction, which is useful for fMRI processing. And it uses a different generative model than EDI, but it's still a Gaussian process. Another tool that we are planning to include in the pipeline is Profumo, FSL's probabilistic functional modes, which is a hierarchical Bayesian modeling of brain networks in big populations using fMRI to characterize patterns of brain activity. It uses a top-down regularization at subject level and bottom-up at group level. The hierarchy is defined over spatial topography and functional connectivity of the modes. And it was developed by Resvan Farahi-Bosorg. And she developed also a stochastic version that allows us to scale to model big data. And it will allow us to generate better fMRI-derived IDPs. So what else? We want to include many updates in the pipeline. For example, fMRI projected into the surface in the way that HCP does. Dynamic models, better diffusion cleaning, improved EDI, improved NODI models, using extract for tractography, segmenting microbleeds from susceptibility-weighted images, improve white matter hyperintensity segmentations with the latest tools, and also, if possible, fully automated QC. So now, apart from all that, we would like to have collaborations. We would like people to suggest solidly established methods. We will evaluate these suggestions with different experts in each area, and we would like this suggestion to match this criteria, the FAST criteria: feasible, aligned, and suitable and testable. Feasible means that we would like tools that are easy to implement, free to use, and not very intensive in terms of memory, disk, or CPU. Regarding alignment with UK Biobank data and goals, we will consider suggestions that are relevant for the UK Biobank data or for datasets that are close to the UK Biobank protocol. Regarding suitable for framework, we only would like to receive suggestions for tools that would fit in the framework, because if we receive any suggestion for tools written in Matlab, for example, they would make the pipeline too complex, create too many dependencies, and if we want to create a containerized version of the pipeline, it will be too big. And finally, testable. We would like to receive with the proposal a way to evaluate, to test the tool or the method clearly described with measurable criteria, and to compare it with other similar tools. If you want to collaborate, send me an email to falmagro@femrid.ox.ac.uk. We will send you a survey to assess if your proposal matches this criteria. And I will be around poster 2260 from tomorrow if you want to ask questions or have a chat about it. Thank you for your attention. And that's it.
Thank you very much, Fidel. Shall we have one question from the audience? Okay, I have other questions from the app as well. So Dr. Fidel, could you give an example about how the UK Biobank brain image pipeline to be sustainable?
Can you repeat that? Okay.
Could you give an example or the feature in the pipeline that will help the whole process, the whole pipeline to be sustainable?
Oh,
Yeah.
well, sustainable in time or environmentally? I don't understand. Well, if it means environmentally, I think that having a central processing of UK Biobank is the way to be environmentally sustainable, because that way we prevent many different labs to do the same processing on the data. If I don't know if they were referring to that question, to that.
Yeah, well, you're welcome to interact with the speaker afterwards. Okay, so thank you again.
Thank you.
And let's proceed with next